Okay, good morning, everybody. Let's continue talking about proto-oncogenes. So remember that we were learning about how viruses, the viruses that are linked to the transformation of cells and eventually linked to different types of cancers. Early on, researchers realized that these viruses were transforming cells because they had certain extra pieces of genetic information that were being carried around as part of their genome that the closely related non-tumorigenic versions of these viruses did not have. And when researchers realized there were these extra bits of genetic information, they suddenly came to the conclusion that these bits must be oncogenes, right? Viral oncogenes that if they are part of an infected cell can lead to transformation. And we learned that that can happen if when a viruses infect a genome of a host cell and different parts of the viral genome integrate randomly, every so often, just by sheer chance, the viral provirus, right, the genome of the virus might integrate right next to something like a normal cell cycle gene in the genome, and it'll pick it up as that virus then replicates, right? It's kind of messy. So not only does it replicate its own genetic information, but there's some sloppiness to it. So it might pick up a bit of extra genetic material as well. And most of the time, that extra bit of genetic material probably is just junk, right? So it may not even have an impact on this virus and how it functions. But every so often, it might pick up, just by sheer coincidence, a functional gene. And if that gene turns out to be something that then is something that controls the cell cycle, and it becomes mutated in that virus, then maybe when the virus infects another host cell, having that mutant form of this gene expressed can stimulate transformation. Right. And we said that researchers realized, though, that many of these examples of such extra bits of the host genome represented normal genes that were borrowed or stolen that way by the virus. Right. And so we now call those proto-oncogenes. In other words, the normal genes in our genome that serve important functions related to things like cell cycle, but are normally behaving well, right? So our cells depend upon them and our cells control them very carefully to control the cell cycle. So that's why they have the potential, though, if they get messed up, to become no longer proto-oncogenes, but full-blown oncogenes. So today we're going to look at how does that happen, right? And how do these proto-oncogenes sometimes become activated and become oncogenic? Okay. So just a bit of recap, key concepts. We discussed that viruses likely acquired oncogenes such as VSARC from fragments of normal host proto-oncogenes like CSARC. Right? So these viral fragments that integrated as fragments can actually impact the proper expression of host genes. So proto-oncogenes such as SARC and MYC that are a normal part of our genome normally control the activity of many other genes and proteins. Thus, the potential loss of regulation of something like SARC and MYC, right, actually has a pronounced effect. So hopefully this makes sense, that if the gene you're talking about that's becoming altered is a gene that's very important because it controls the behavior of many downstream genes, right, then having a mutation or an alteration in that first gene might have a more pronounced effect on something like transformation happening. So this is kind of like, I think of it this way. It's like if you have a busy factory floor where, in this case, maybe this company is making certain types of machines or equipment. Right? And so you have these different stations, or you have the different skilled workers, like these guys here, working on assembling the machine, right? So they have a very specific knowledge set, right? They know how to put together these particular pieces of the machine. And then maybe other coworkers know how to put together other parts, right? So then you have this guy over here. He's like the floor supervisor, right? He comes around and he makes sure that everything is going correctly, that the products that they're making, meet the expected requirements, right? So the supervisor then, he's kind of more higher up, if you want to think about it that way, right? Because changes in his decision making, like if he says, hey, we need to go ahead and make more of this piece of equipment compared to this piece of equipment. So you guys stop doing that, go over there, or you need to speed up. You're kind of like, slowing everything down, right? So he is a supervisor. And so changes in his behavior might have a broader effect on the overall functioning of the factory compared to if you just had one of these workers like this guy here, right? If he suddenly decided he wanted to try making, a different technique for making this thing, any changes would be sort of more limited, right, to just that particular aspect of the factory's production output. Okay? So that's why I want you to think about this example here. What happens when an individual worker versus a supervisor changes behavior? And if I were to ask you then, in that case, when we talk about proto-oncogenes such as SARC and MYC, right, hopefully what you realize is we're talking about these genes play a more important higher up role in controlling the activity of the cell. So a question that was faced by early oncologists was that, they realized not all cancers are due to viral infection, right? We said something like 1/5 of all cancer types have strong causative links with viruses. And yet, in cancer, you have these proto-oncogenes like SARC and MYC that start off as well-behaved proto-oncogenes. I always stress to the students, because students, it's easy to say like, wow, evolutionarily, if these things are dangerous because if they mutate, they cause cancer, why do we even have them, right? But the whole point that I try to emphasize is they are critical genes because these are genes that control major functions of things like cell proliferation. So if you kind of flip that around, it makes sense then that these would be the genes that if they start going wrong or bad, or your cell loses control of them, you start having more pronounced effects. And they become oncogenic, right? The functions of these altered genes. But if it's not from viral infection, then how do proto-oncogenes become oncogenes in otherwise normal cells? So early on, there were two sort of competing theories about this. One was that some researchers thought, well, there must be these endogenous latent retroviruses that are kind of hanging out in the human genome. So even though you're no longer experiencing a viral infection, right, maybe, through the course of evolution, there are bits and pieces of viral DNA that are part of our genome. And every so often, maybe one of those latent kind of sleeping viral genomes could wake up and cause like a very localized infection just within that cell in a way, right? That would cause things like, viral oncogenes to form. The other competing theory was that the proto-oncogenes in our genome, they become altered not because of viral activity, but they become altered due to mutations or things like changes in expression level, amplification or silencing, right? So let's take a look at the first theory. Are there endogenous proviruses hidden in our genome? What do you think about that, right? Our genome is a pretty big place, right? 3.2 billion base pairs of DNA, and, most of it is not coding for genes. It does turn out that there are viral fragments in our genome, but none of these are complete proviral fragments. Right? I think one way to think about it is if there were large intact proviral genomes in our genome, that probably would have been gotten rid of by natural selection, right? Because those genomes then would always be susceptible to this idea of a virus waking up and suddenly spontaneously being made from within your own cells. What a weird thought that is, right? Because we're always thinking about viruses coming from the outside and infecting us and making our cells make new virus particles, right? What if there were, in fact, these hidden viral genomes carried in our human genome that can spontaneously wake up and you start just making virus particles on your own? Yeah, you can see that would not be allowed. Natural selection would not favor that, right? So we don't have large intact pieces of viral genomes, but we certainly do have many fragments of sort of useless viral bits and pieces that are carried around in our genome, right? So that may surprise you, right? That we do in fact have this baggage, right? Because remember, most of the time, or a lot of the time, I don't know if most of the time, when viruses infect cells and they're randomly kind of integrating fragments in them, right, that virus will only succeed in hijacking the cell if it succeeds in getting its genome sort of stuck in a place where it can be expressed. And then the cell makes new virus particles, right? And then the cell blows up essentially. So end of story, right? But what about all those cases where you have bits and pieces of the viral genomes that just became part of a genome that was then passed on in germline cells, right? Because germline cells can certainly be infected by virus particles as well. So maybe it's not surprising then that over millions and millions of years, right, we ended up actually carrying with us bits and pieces of these viral particles. Bits and pieces of the viral genome, but none of these can make new viruses. How much of our genome are actually viral in origin? This may surprise you, right? As much as 8% of the human genome is actually of viral origin. And that goes a long way in explaining why, right, our physical genome is 3.2 billion base pairs, but per chromosome, right, only a small percentage of that sequence is actually coding DNA. Right? The rest of it is things like regulatory sites, and 8% of it is actually of viral origin, or different repeat regions that are sort of these nonsensical repeats of certain nucleotide patterns over and over and over again. All right. So that pretty much eliminates the first theory then. So then the focus became on this idea of proto-oncogenes like c-Sark, c-Myc in our cells somehow become altered due to mutation or amplification. Right? How can we identify proto-oncogenes if they are in fact just normal wild type genes that really kind of look like any other gene? Right? So one key approach has been what I've been teaching you. Remember, early researchers, when they looked at Rous sarcoma virus, the one that caused chicken sarcoma tumors, right, and compared it to the closely related avian leukosis virus. They're both bird viruses. They're both very similar. They have very similar genomes, but the ALV, the avian leukosis virus, does not cause cells to transform, but the Rous sarcoma virus does. That's when they identified the Sark region of it, the extra piece of chicken or bird genome that got picked up by the virus, and then it mutated in some way and became what they called VSARC, right? Viral Sark, right? And then they found that actually in a normal healthy cell, there is Sark already, right? And they called that the c-Sark. So if you take that sort of little story, you realize that one way you can identify certain genes that are normally part of the genome that are relevant for things like cell proliferation or different types of qualities that we associate with things like cancer if they go bad, is to look at these things that are found in the viruses. So one approach, okay, would be to, for example, identify overrepresented fragments of DNA in tumor cells. Right? So you could say, well, I have this tumor here. I'm going to analyze the genome of these tumor cells. I'm aware that oftentimes you have gross amplification of certain genes. Remember, we looked at karyotypes where there were like 3 or 4 chromosome 1s, and different regions were all different colored in the karyotype because it's all mixed up with other chromosomes. So that can certainly lead to overexpression of certain proteins, and maybe those proteins are the ones that are important in cancer. So therefore, we can try to identify what fragments of DNA are overrepresented in tumor cells, and then try to map those back to the genome to identify the context, right, of where they originated from. So it's almost like using the find tool in a text document, right? If you have a large Word document and you're trying to find one particular phrase or sentence in it, right? These days, we all know how to use the find tool of something like Microsoft Word. So you can do a similar approach to find important proto-oncogenes. So early experiments dealt with, sort of a brute force kind of way of doing this. They would grow cultures of tumor cells, so at low magnification. This is a culture of mouse fibroblasts. So these are NIH3T3 cells. These were transfected with human DNA from bladder carcinoma cells. So in other words, they took the DNA from a human tumor, right? And the assumption is that DNA is messed up in some way, right? So as that's the source of why the tumor formed in the first place. And researchers said, well, let's go ahead and stick that DNA into these mouse fibroblast cells and see what happens, right? And sure enough, certain populations of these fibroblasts would then transform, okay, and they would start turning precancerous, right? So you'd have these concentrated foci of cells that are beginning to bunch up on each other, right? So recall, we talked about many of the morphologic changes, the phenotypic changes of these cells no longer being nicely spaced apart, right? So these are normal cells on the boundaries here. They become very bunched up together. You no longer have the contact inhibition, and in fact, they can start piling up. Right? So the normal cells have the normal morphology. They're nice and flat, almost like little cobblestones here. Right, that's what these cells are. Whereas if you look at the transformed cells, yeah, all right, they're all bunched up and a real mess. So what they did was they would go ahead and they would transform mouse fibroblasts that way, and then they wanted to ask the question, okay, so in these transformed fibroblast cells, clearly they must contain the particular DNA sequences that are responsible for their transformation, right? So they would extract the DNA from them, and then they would go ahead and bust up the DNA into small fragments. Right? They'd bust the DNA into small fragments, and hopefully some of these fragments would actually have whatever gene is responsible here for causing this. Right? They would then go back to the normal mouse fibroblasts and put those little pieces of DNA on there, and they would see, oh, right, so some of these cells got whatever little fragments were required to transform them. So then they would take those cells and they would inject them into a mouse model to see what would happen, right? Because clearly these were mouse fibroblast cells to start with. So whatever these fragments of DNA were that caused transformation, they're capable of causing transformation of mouse cells. So they injected those, and then they would see that the mouse would then develop a tumor, right? And then along the way, they would try to do things like analyze the DNA fragments and try to figure out what are these different sequences of these fragments that were allowing the transformation and tumorigenesis to occur. So this was done, way back in the 70s, well before we had things like PCR, even well before we had computer-assisted alignment tools of sequences with genomes, right? So with PCR in the 80s, that really revolutionized our ability to work with small fragments of DNA. And you wouldn't have to necessarily go through this big laborious process of actually trying to figure out the sequences of little fragments and then methodically trying to map them back. But the concept here is this one of using the find tool in your word processing software. Let's say, for example, right, that you're trying to find a particular phrase in the complete works of William Shakespeare, right? So let's think of an example of a famous Shakespearean soliloquy, right? A phrase that you associate with Shakespeare. Okay, so you might say, well, when you think of Shakespeare, you think of something like, oh, to be or not to be, because that's probably the most famous quote that everybody knows. But you know that's Shakespeare. But do you actually know where does that come from in Shakespeare's works? What is the meaning of to be or not to be, right? Because if you just have that little phrase out of context, it just becomes like a pop culture kind of thing, right? To be or not to be. But do you actually understand anything about the context of its meaning and, importantly, how it fits in with what's going on in the particular work that Shakespeare, first used that phrase in, right? So here you have the complete works of Shakespeare, which is like 884, 647 words, 884,647 words. How are you going to find to be or not to be in here, right? If you can't use the internet, what can you do? So this is where you can use the find tool. So let's say you're given the complete works of Shakespeare as a humongous Word document. Right? So in this case, we're saying the complete works of Shakespeare, all of his plays and things like that. Think of that as the genome, right? And maybe the different plays are different chromosomes, and each play is made up of several tens of thousands of words, right? Sequences that make sense, that contain information in the context of that play. So if you use the find tool to search for a famous quote, right, then you get a hit. You get a hit pretty quickly. And now you know, oh, to be or not to be is from Hamlet, Act 3, Scene 1, right? So, and then if you read this, to be or not to be, here's the whole soliloquy. And then you realize, because now you know the context, right? You know it's from Hamlet, you know it's from this particular scene, then you can read, right, the scene that surrounds it and understand what to be or not to be is talking about, right? So in this case, it's Hamlet going off on this, kind of existential kind of crisis. So here's a painting, so here's Ophelia, Princess Ophelia, and here's her loser boyfriend, right, who's all emo. He's always to be or not to be. Anyhow, so you find the context of to be or not to be, and it's part of the soliloquy, and it's 283 words total. Now you have the ability, right, to be able to know something meaningful about this sequence, right? Not just that it stands out as something that's an obvious Shakespeare quote, just like a viral oncogene, right? You can identify a short phrase, the sequence of a viral oncogene, but only once you put it in context in the genome where it came from, do you suddenly have a sense of why it's important and how you can begin to appreciate what this gene does. So these are examples of retrovirus-associated oncogenes that have been discovered by looking at different human cancers, right, finding that suspect kind of sequence that's part of the particular virus, right, and then mapping it back, sort of using the find tool to figure out where it is in the genome. And then researchers then, not surprisingly, would pay close attention to understanding where it is on the chromosome and the context, the surrounding sequences that may involve other genes or other regulatory sequences, right? So that's how things like SARC was discovered. Other important proto-oncogenes like RAF, ERB, ABL, the RAS genes, MYC, for example. So these all represent endogenous proto-oncogenes that may become oncogenes through amplification or gene mutations, or even being borrowed by a virus and having the mutation happen in the virus before the virus then infects a cell and brings the mutated version back into the cell. All right, so let's look at some examples. Let's start with this ERB. All right, so it was comes from avian erythroblastosis. So it was first identified in bird viruses, but they went ahead and identified it in the genome of the mouse, right? When they looked at a mammal model, they looked at the mouse and they named it ERB2. Okay, and it is a receptor tyrosine kinase. We will talk in a later lecture about the importance of these tyrosine kinases. I think I already mentioned to you that that's a very important type of receptor protein. And it affects things like gastric, lung, and breast cancers. We can also look at another example of Harvey sarcoma. All right, so Harvey sarcoma was identified in mouse and rats. And the gene that's been identified is this RAS gene, right? And because it's named after the rat sarcomas that they were identified in. And this is not a receptor, this is a small G protein, and this is involved in bladder cancers. All right, so two examples of different gene products that are very important. So let's take a look at the importance of this. So ERB2 is also known by its alternate gene names of HER2 and NU. Remember, different researchers independently oftentimes discover the same gene when they're looking at things like, cancer, different diseases, and they will name them independently. And then it turns out, after these papers are published, that suddenly people realize, oh, this is actually the same gene that these separate groups identified. But a lot of times, it becomes like kind of this like turf war of whose name that they gave this gene is the one that actually sticks, right? So a lot of times there is no clear winner and they have multiple names. So ERB2 is the same thing as HER2 and is the same thing as NU. And this is a gene that's highly implicated in certain gynecologic cancers such as breast cancer. In this case here, we have some clinical samples, some clinical data where they've taken 5 different tumors from 5 different patients. Okay? And these patients, their tumors are exhibiting different degrees of aggressiveness, right? Different extents to which these tumors are rapidly dividing. And so the researchers first took a look at the DNA, right, of, in particular, this ERB2 HER2 NU gene. Okay? So they did a southern blot to actually look at, these bands, the intensity of these bands just represent how much of the particular DNA, the ERB2 HER2 NU region of the DNA, is present in these samples. Keep in mind, right, these bands don't come from one cell. They're collecting pieces of tissue that have millions of cells, right, and then getting all the DNA out of them. And so when you then run a southern blot, this is the combined intensity representing how much DNA of this particular gene is present in this population of cells that you've purified the DNA from. That's kind of important to keep in mind, right? But of course, they normalize it so that the total amount of DNA, of genomic DNA from sample 1 to sample 5 is the same amount of total DNA, right? So that way, the difference is in intensity of just the ERB2 HER2 fragment of DNA, right? Makes sense when you compare across the 5 different samples. So you can see here in patient 1, there's very clear evidence of over amplification of the gene itself. So maybe many copies of the ERB2 HER2 gene in this person's tumor cell genome, right? Maybe let's say, if just for argument's sake, let's say patient 3, this is considered the normal expression level, not expression level, sorry, the normal copy number of how many copies of ERB2 HER2 gene there are in the genome, right? So in some cases, you actually have, maybe some evidence of underexpression, right? So 4 and 5, look very similar, right? If you then said, well, that's got to explain it then, right? If you have multiple copies of a gene, then very clearly you're going to overexpress the gene, end of story, right? But it's not that simple, because remember, gene expression going from DNA all the way eventually to a functional protein, there are many steps in between, right? We learn about this in, for example, Bio 275, right? You can control the transcription rate of things like RNA Pol II on the gene. You can control how the RNA transcript gets edited, right, to make mRNA And you can also control how that mRNA gets translated by the ribosomes into the finished protein. Right? So just starting with more copies of the gene than normal in itself doesn't translate, one to one with having more protein. And you can see here, certainly, for example, in patient number 1, having many copies of the gene does seem to lead to a lot of RNA, right? So this is like, I don't know if this is primary RNA. I think it's maybe messenger RNA, right? Lots of RNA. So that's kind of obvious, okay? But look at something like patient 5. Here you have more or less normal copy number of the ERB2/HER2, but yet, right, you get a ton of RNA being made, almost as much as in patient 1, right? So this might lead you to believe, for example, right, that it's nothing wrong necessarily with the ERB2/HER2 gene itself. Maybe there's something wrong with one of the intermediate factors in the process of transcribing, right, the DNA into RNA has somehow amplified the amount of RNA that's being made, right? Then the third level then would be after it's translated by the ribosomes, how much actual ERB2/HER2 new protein do you have at the end? And you can see rather surprisingly, even though patient 1 had a ton of RNA made, right, the relative amount of RNA being made, right, is actually looks like it's just as much of protein being made at the end, right? It looks like about just as much as patient 5, right? So when you actually look at the tumors from these patients under a pathology, in the pathology clinic, right, when they stain these sections of the tissue, the tumor tissue, with antibodies that will detect the presence of the ERB2/HER2 protein, right? Sure enough, you can see the cells stain very dark brown with patient 1 and patient 5, right? Maybe we said patient 3 just for argument's sake, right? Maybe this is the normal expression level of ERB2/HER2. And in patient 4, right, it looks like there's very little of it. There's so little of it that in the image here, they had to increase the contrast of the background just to try to get to visualize, right? So the take-home message here is, yes, gene amplification itself, having multiple copies of a gene as a result of the integrity of the human genome being compromised, sure, that can lead to amplification, but clearly there's more to it than just that, right? The involvement of other genes must be looked at because they can explain how, way more RNA is made, even though you're starting off with the right number of gene copies, right? And there are other genes involved then in determining how the translation process happens, right? So this is what I mean by understanding context, right? Just identifying a gene as being important is not enough. You have to build up a picture of how that gene is dependent upon other genes to actually try to explain and connect the dots as to how this is actually leading to cancer. All right. Oh, so by the way, ERB2 was originally named after its protein size. So this is something we'll see a lot of this semester. So a lot of times researchers who isolate a particular protein, it's in the context of a whole bunch of other proteins that they're isolating. Maybe at that point in our scientific knowledge, they did not know the identity of these different proteins, but they wanted to say, yeah, some of these proteins I think are really important because we got these from a tumor cell or tumor tissue and they're overexpressed, right? And so they would just name them temporarily based upon their size. So P185 is because this protein migrates on a polyacrylamide gel at 185 kilodaltons, right? So then later, people who study the P185 protein here, right, they're the ones who then called it something like ERB2 or HER2 or nu, once they sort of determined, yeah, this thing is important, and we want to identify it as a oncogene, right? In some cases, that simple nomenclature of P185 is still used to this day as the preferred name of some of these proteins. So for example, we will learn about P53 for example, a very important tumor suppressor protein, right? That is just known as p53. Okay. So here, this is the first time I'm showing you this type of data. This type of data is very important for you to get familiar with because it represents a common way of reporting clinical data. Right? This is what we call a Kaplan-Meier plot. A Kaplan-Meier plot is mainly used in the clinical setting, where you have a group of patients who you're sort of treating as one or more cohorts, right? Group of different patients that you're following based upon some variable that's different between the different groups. Right? And the Kaplan-Meier plot follows these patients over time until a certain outcome that you've chosen beforehand, right, that you want to say is the outcome that you're trying to follow. When that outcome happens, that's when the Kaplan-Meier plot, you would have a data point. So in this Kaplan-Meier plot, we were looking at a group of breast cancer patients, right? And the researchers asked the question, when these patients first presented with the disease and were diagnosed in the clinic and biopsies were taken of the tumors, right? These researchers were interested in finding out, does HER2/neu protein levels really serve as some sort of prognostic indicator of how this patient's going to do, right? So they were then, these slides were sent to the pathology lab, and they did those different types of data analysis that I showed you on the previous slide, looking at copy number, RNA, and also staining the tissue to look at the presence of HER2 and neu. And they divided the set of patient samples into two groups then, right? In some patients, HER2/neu was not amplified. So there were 52 patients where they didn't see any evidence of the HER2/neu being amplified, right? So that might have been like patient 3 or something on the previous slide that we said, for argument's sake, was considered normal, right? Then you had 11 patients who had HER2/neu amplified. And in this case, you know, to be specific, this data is just saying, well, if they had more than 5 copies of the gene present, right? So now they're just looking at copy number. And then these patients were then clinically treated, right? And followed over time. How many months after that initial treatment could they remain disease-free? In other words, the outcome in this case is relapse, right? When the tumor returns, that's the endpoint of that particular patient, and that would be a data point, right? So here on the y-axis then is the proportion of these groups of patients who remain disease-free at these different time points. And so all of them start at 100%, right? Because that's the starting point. And then you can see over time, every time a patient came back into the clinic and there was recurrence of the tumor, right, they would drop down. That group would drop down, you know, based upon the fact that a patient has now had relapse, right? And what you can see is that the two groups, the HER2/neu not amplified versus the HER2/neu amplified, right, very clearly there is a significant difference in the length of time a patient could be expected to go disease-free, right, following diagnosis, depending upon whether that tumor that was first diagnosed already had HER2/neu amplified or not. Right? So Kaplan-Meier plots, you can see why this is useful, right? Because you can suddenly get an important picture here that HER2/neu might be a very useful marker for prognosis. So now you can say when a patient presents with breast cancer, maybe one of the first tests you do is look at HER2/neu overexpression, right? Because that can already give you some sense of time frame. You'll see a lot of Kaplan-Meier plots, not only in our lectures, but also in the articles that you read. So it's important that you get familiar with them. Another way you can look at data, a much more contemporary way, you know, because people have been doing Kaplan-Meier plots for like decades, right? Because you can pretty easily just follow the outcome data in a clinic that way. But more fancier modern ways of analysis do things like microexpression array data, right? So here, what we're doing is we're going to look at levels of ERB2 HER2 expression based upon not gene copy number, but the next step, looking at how much RNA is actually present in tumors, right? The RNA that ends up being translated as ERB2 and HER2 protein. Okay. So what they've done here is they've taken 360 different primary breast tumors from 360 different patients, right? Primary breast tumor means this is the first tumor that appeared and is what maybe was detected as a lump, right? And that's what made the patient go to the clinic and a biopsy was taken. And what the researchers have done is they already want to ask the question, does ERB2 HER2 expression level have something to do, right, with things like the fact that these tumors were caused by altered expression level? And so they know where ERB2 and HER2, the gene, is. It's located on chromosome 17, and in particular, the Q arm of 17, 17q12, right? So here's chromosome 17 here. So 17q12 is around here, right? This is where the ERB2 HER2 gene is. So what they can do is they can design a whole bunch of little probes, right? Probes that can match up with RNA that's coming from any of the genes or any of the genetic sequences that are being expressed in this region of the chromosome. So probes not only that are very specific to the ERB2 gene and the ERB2 RNA, but you can canvass the entire area upstream and downstream with probes as well, just to see if there's other RNA messages coming off of those, right? So then what you do is the computer, right, will do this analysis and it will label the expression level of RNA that it detected in this kind of a heat map type of pattern, right? Green meaning normal levels of RNA expression. And if there's higher than normal levels of RNA expression, then you would go more towards red, right? So that means each one of these pixels on this grid here, so it's 360 columns wide, right? Each column going down then, right, is one tumor sample. And each row going across would be a different probe designed to literally cover or canvas the entire region around the 17q12. Okay. Here they set it up so that the ERB2 HER2 RNA probes would be right in the middle here. So that means each little column, you can imagine, because it's a different patient, will be a different sort of order of green dots, red dots, green dots, red dots going down, right? And if you did not organize that, if you just looked at the data, this whole thing would just look like a big mess, right? Because it would just be green dots mixed in with red dots everywhere, right? Because it would just be randomly lined up, these columns, these 360 different patients. But this is the cool part. You can then ask the software to actually start doing this process of taking one column of green and red dots, right, and comparing it with the other 359 columns of green and red dots from the other patients, and try to find the next column that would match up closest in pattern of green and red to the first one. And then the software can keep doing that. So it's grouping. It's deciding how to line up the 360 patients based upon similarities in the green and red dot pattern going down, right? Each one of these hundreds of little probes going down. And that's how you arrive at this type of microarray data, right? So what it's done is the software has grouped them together. And what you can see very clearly is there is a subset of patients, right? This is maybe like, maybe like a fourth. So this is like maybe 90 patients over here, right? Who very clearly have elevated RNA expression levels of ERB2 as part of their tumor. Right? Whereas you have a big group of patients over here, like the more left that you go, where there's decreasing evidence that ERB2 and HER2 was somehow linked to their particular tumor, right? Because they have mostly green in this region, normal expression level. Right? But what's really neat about this type of data is not only can you tell the ERB2 tumors, the tumors that maybe had ERB2 be responsible versus the ones that didn't, but look at this. You can also identify other groups of patients. So this represents a group of patients here, maybe the 90 or so, whose data are in these columns, who don't necessarily have ERB2 overexpression, but they have overexpression of this region of 17q12, right? And that maps to these genes over here. So you can see how this type of analysis really opens the door to not only confirming certain things and seeing what types of genotypes may be responsible for a patient's phenotype, but opening your eyes to other possible hypotheses that you may have missed otherwise, right? So that means these researchers now may say, we need to take a look at this GSDML or this ORML-DL3 genes, because clearly it looks like that is responsible, that overexpression is responsible for some patients' tumors. Okay. So let's finish up with this discussion of the other example that I gave, which are RAS families of proto-oncogenes. Okay. So they were originally described in rat sarcomas in the 1960s. And the human genome, we actually have 3 copies of this RAS gene on 3 different chromosomes. So NRAS is on chromosome 1, HRAS is on chromosome 11, KRAS is on chromosome 12. All 3 of these genes are nearly identical in sequence. They are 189 amino acid proteins, right? But there are variations in some amino acids in them, right? So what probably happened is that sometime in ancient mammalian evolution, there was one RAS gene that then got duplicated, right? And became part of another chromosome. And it actually turned out to be useful to have 2 different RAS genes on 2 different chromosomes. And over evolutionary time, they got different mutations, right? Polymorphisms, whatever, that made them functionally, very similar RAS genes, but important in different pathways. And then it happened again, you have a third one. Evidence that they are relevant in different pathways is the fact that the N, the H, and the K represent the disease that this RAS gene was associated with. So neuroblastoma, so the RAS that's on chromosome 1 is linked to neuroblastoma. The one on 11 is Harvey sarcoma. The one on chromosome 12, when it gets mutated and changes, leads to a different type of sarcoma, Kirsten sarcoma. Right? RAS is super important in cancer progression. Around 25% of all cancers involve some kind of a RAS mutation, right? And in some types of cancers, up to 90% of the tumors that you study of that particular cancer, you will find a RAS mutation. Okay. So this is a list of different types of tumors, different cancers, and the proportion of tumors that carry a particular mutated RAS gene or are associated with a particular mutation, right? So 90% of pancreatic cancers have a mutation in the KRAS gene, right? Whereas something like in thyroid cancers, 60% of the thyroid cancer cases, you can identify a mutation in either HRAS, KRAS, or NRAS, okay, et cetera, et cetera. Again, don't memorize this, but understand what I'm trying to show you here, right? That these RAS genes are a very important proto-oncogene because when they get mutated, right, they have a very important role in the development of cancer. We will study, what they actually do in the next lecture, but the fact that you have 3 of them, right, and they're slightly different as a result of evolutionary time, getting slight different polymorphisms and their sequences being altered, they are relevant to different pathways in different cells. Right? Okay, so this is the last slide for today. Let's talk about these mutations. Here we have the amino acid number identifying the sequence of HRAS. So amino acid number 1, it's a methionine, right? So the ATG. But then you can see you go along the the first part of the protein, and this is a splice point, right? So this would now be an intron. So this would then be joined up with the next exon, right? And the amino acid sequence, you can continue on. But it's okay if we stop there, because amino acid 12 in HRAS normally has a GGC in the DNA. That's the codon, and it codes for glycine. So the normal wild type, well-behaved HRAS proto-oncogene has a glycine in this position, right? But if this is mutated, so it's a GTC now, right? That codes for valine, and this is the oncogenic form of the protein, right? So sorry, I lied. I realize this is the second part of that slide. If you were to look at, let's say, 10,000 tumors, right, that have a RAS mutation, in this case, KRAS, and you were to actually map out what are the different mutations, the different amino acid substitutions that you see in these tumors, there's a really striking finding. What you find is that here is amino acid 1, and if you were to map this out from the N terminus all the way to the C terminus of the protein, right, you'd find that, you know, more or less mutations in these other regions don't show up that much in these tumors, right? But look at this. Practically all of the tumors, you know, out of 10,000 or so, right, have an amino acid substitution at that amino acid 12 position, right? Then the second highest one is tumors have a mutation at amino acid 13. Then the third one is you see this amino acid substitution at 61, right? So this is what I want you to think about, and I'm sorry that I've gone over. Mutations happen randomly, right? So there's no reason to believe that the codon that codes for amino acid 12 or 13 or 61 are any more or less likely to be mutated than one that codes at position 120 or 160, right? So why do you get data that looks like this, that shows that the overwhelming majority, right, of these mutations are being seen at position 12? That's where I'll stop today, because I want you to think about this, and we will continue this discussion on Friday. Okay? All right. Let me stop.